Scientists unveil a more diverse human genome. The pangenome, which collated genetic sequences from 47 people of diverse ethnic backgrounds, could greatly expand the reach of personalized medicine. More than 20 years after scientists first released a draft sequence of the human genome, the Book of Life has been given a long overdue rewrite. A more accurate and inclusive edition of our genetic code was published on Wednesday, marking a major step toward a deeper understanding of human biology and personalized medicine for people from a wide range of racial and ethnic backgrounds. Unlike the previous reference, which was largely based on the DNA of one mixed-race man from Buffalo, with inputs from a few dozen other individuals, mostly of European descent, the new pangenome incorporates near-complete genetic sequences from 47 men and women of diverse origins, including African Americans, Caribbean Islanders, East Asians, West Africans, and South Americans. The revamped genome map represents a crucial tool for scientists and clinicians hoping to identify genetic variations associated with disease. It also promises to deliver treatments that can benefit all people, regardless of their race, ethnicity, or ancestry, researchers said. It's been long needed, and they've done a very good job, said Ewan Burney, a geneticist and Deputy Director General of the European Molecular Biology Laboratory, who was not involved in the effort. This will improve our fine-grained understanding of variation, and then that research will open new opportunities toward clinical applications. Powered by the latest in DNA sequencing technology, the pangenome collates all 47 unique genomes into a single resource, providing the most detailed picture yet of the code that powers our cells. Gaps in the earlier reference are now filled, with nearly 120 million previously missing DNA letters added to the 3 billion letter long code. Gone is the idea of a totemic strand of DNA that extends 6 feet when uncoiled and stretched out in a straight line. Now, the rebooted reference resembles a corn maze with alternative paths and side trails that allow scientists to explore a broader range of the genetic diversity found in people the world over. The new pangenome reference shows many possible routes for a sequence to take, represented by the different colors. Yellow is a duplication variant. Pink is an inversion variant, green and blue represent a deletion variant, and light blue is an insertion variant. Credit Daryl Leyengree. Eric Green, director of the National Human Genome Research Institute, the government agency that funded the work, likens the pangenome to a new kind of bodywork manual for automotive repair shops. Whereas before, every mechanic only had the design specs for one kind of car, now there is a master plan that covers different makes and models. We've gone from having one really nice blueprint of the Chevy to now having blueprints of 47 representative cars from each of 47 different manufacturers, he said. Knowing what to do with this Kelly Blue Book of Genomics will involve a steep learning curve. New analytical tools are needed. Coordinate systems must be redefined. Widespread adoption will take time. Making this easy to be used by the community is work to be done, said Heidi Ram, Chief Genomics Officer at Massachusetts General Hospital in Boston, who was not involved in the project. But in due course, experts said, the pangenome will revolutionize the field of genomic medicine. We are going to have the benefit of actually understanding ourselves as a species much better, said Evan Eichler, a genome scientist at the University of Washington, D.R. Eichler was among more than 100 scientists and bioethicists who described the new pangenome reference in the journal Nature. The architects of the project are continuing to add more population groups with the goal of including at least 350 high-quality genomes that encompass the bulk of global human diversity. We want to represent all the branches of the human tree, said Ira Hall, a geneticist who leads the Yale Center for Genomic Health. Some of the new genomes will come from New Yorkers who previously participated in a research program at the Mount Sinai Health System. If their preliminary DNA data seems to reflect certain underrepresented genetic backgrounds, those individuals will be invited to participate in the pangenome project. Some gaps might never get plugged in the publicly available reference, though, by design. Previous attempts to capture human genetic diversity often extracted sequence data from marginalized populations without regard for their needs and preferences. Informed by those ethical missteps, pangenome coordinators are now collaborating with indigenous groups to develop formal policies around data ownership. We are still grappling with the issue of native and tribal sovereignty, said Barbara Koenig, a bioethicist at the University of California, San Francisco, who was involved in the project. In Australia, researchers are incorporating DNA sequences from various Aboriginal peoples into a similar depository that will be combined with the open-source pangenome, but then kept behind a firewall. According to Hardeep Patel of Australia's National Centre for Indigenous Genomics in Canberra, the scientists next plan to consult with community leaders about if or how to make the data accessible through request. Some Indigenous advocates want to see the pangenome project go further. Keolu Fox, a genomics scientist at the University of California, San Diego, who is native Hawaiian, has suggested training the next generation of indigenous scientists to have greater agency over the genomic data. It's finally time that we decentralize power and control and redistribute that among the communities themselves, Dr. Fox said. Genome analysis shows humans survive on low number of genes, opening a new era in human biology and medicine. Two rival teams of scientists this week present their first interpretations of the human genome, the set of DNA encoded instructions that specify a person. The two teams report in articles to be published on Thursday and Friday that there are far fewer human genes than thought, probably a mere 30,000 or so, only a third more than those found in the roundworm. One team, Celera Genomics, has compiled a parts list of the proteins needed to make a person. The other team, a publicly funded consortium, has traced the history of how the junk regions of the genome accumulated and has found that small elements of the junk may play a useful role. They also discovered that human genes have been derived directly from bacteria. The two teams announced last June that they had assembled the human genome, but it has taken them until now to analyze their findings. The interpretation of the genome, identifying the genes, their functions and controls, and how they relate to human physiology and disease, is expected in time to revolutionize medicine by clarifying the mechanism of many diseases and generating new tests and treatments. Physically, the genome is minuscule. 2 copies of it are packed into the nucleus of every ordinary human cell, each one of which is about a fifth the size of the smallest speck of dust the eye can see. But the genome is vast in terms of its informational content. Composed of chemical symbols designated by a 4-letter alphabet of As, T's, C's, and G's, the human genome is some 3.2 billion letters in length. If printed in standard type, it would cover 75,490 pages of this newspaper. The enormous task of decoding the genomic message began in 1990 and is now substantially complete, although both teams' versions of the genome are riddled with gaps. With so much effort and scientific glory at stake, members of each team remain highly critical of the other's approach, believing that their own strategy for decoding the genome is likely to produce the better and more accurate version. Since last June, however, both have been muting criticism and observing a limited truce. The pact called for a joint announcement, made at the White House on June 26 last year, that each side had finished assembling its version of the genome, and for joint publication of their findings, which is occurring later this week. The joint publication, however, is about as separate as a union could be, with each side's articles appearing in rival scientific journals issued on different sides of the Atlantic. The findings were to be announced tomorrow, but the embargo was lifted by the 2 journals after the Observer of London broke it. One team is a consortium of academic centers, mostly in the United States and Britain, but with members in France, Germany, China and Japan. The consortium is financed largely by the National Institutes of Health and the Wellcome Trust of London. Its version of the human genome is described in a 62-page article in Nature, based in London. The principal author is Dr. Eric Lander of the Whitehead Institute in Cambridge, Mass. The other team is led by Dr. J. Craig Venter, president of Celera Genomics in Rockville, MD. Its report appears in a 48-page article in Science based in Washington. Despite the two teams' many differences, they largely agree on their findings about the human genome. Theirs is the first overall look at a genetic document of extraordinary strangeness and complexity. No one expected it to be comprehensible at first glance, and the two teams have so far mapped only the principal features of its terrain. Their principal discovery is how few human genes there seem to be. Textbooks have long pegged the number of human genes at around 100,000, but with the sequence of human DNA units in hand, the two teams have found far fewer than expected. Dr. Venter says he has identified 26,588 protein coding genes for sure and another 12,000 possible genes.